Eu, como o Marchante e a Margarida, sou de 2000 há 10 anos atrás, começamos o curso há 10 anos atrás, e começamos a nossa vida académica aqui no ISA, e hoje eu vou-vos falar sobre polifenóis e a doença de Parkinson, mostrando alguns dos resultados, se está a funcionar, alguns dos resultados da minha tese de doutoramento. Eu peço desculpa porque eu tenho o discurso todo planeado em inglês. Pronto. Sim. Mas senti-me mal, mas próxima vez eu prometo fazer o esforço e o exercício de preparar tudo em português, só que eu tenho medo agora de estar aqui a falar em português e as ideias não fluírem tão facilmente. Vou mostrar muitas técnicas, mas depois estejam à vontade de fazer as perguntas todas que acharem por bem. So, first of all, Parkinson's disease, it's the most common movement disorder. And currently, it does not have an effective therapy. The symptoms observed in Parkinson's disease patients, it's caused by the loss of dopaminergic neurons here at the substantia nigra, as you can see here in this picture. The main pathological hallmark of Parkinson's disease, it's the presence of these cytoplasmatic inclusions called Lewy bodies in these dopaminergic neurons that it's maybe related with cell death. These cytoplasmatic inclusions are mainly composed by misfolded and aggregated alpha-synuclein. What is alpha-synuclein, after all? This is a protein that is intrinsically an unfolded protein because it does not have a stable 3D structure. So, under pathological conditions, it misfolds and aggregates, forming oligomers and then bigger structures as amyloid fibers that are the building blocks for the Lewy bodies, as I explained previously. It is known that these fibers, these oligomers, and even these Lewy bodies are related with oxidative stress, synaptic dysfunction, and impairment of several cellular pathways, leading to toxicity. On the other hand, polyphenols are compounds that occur naturally in plants, although they can also be synthesized, and you may know some of them. For instance, epicatechin from green tea, resveratrol, which is very famous now, and it's found in grapes, and even catechin in cocoa, in chocolate, that we all like. These polyphenols are emerging in human health due to epidemiological, clinical, and even preclinical studies showing their importance in several human diseases, reducing their risk. For instance, gastrointestinal diseases, atherosclerosis, and even cancer and neurological disorders. So the main goal of this study was to study the neuroprotective mechanisms underlying the polyphenols protection in Parkinson's disease. For this, we extracted polyphenols from several plant species, some of them already well studied, as ginkgo biloba and Rubus lideus, and two of them, arbutinib and Corema album, who were still poorly characterized. We extracted the polyphenols from the leaves and the fruits and submitted them to a solid phase extraction to obtain the polyphenol enriched fraction, or PEFs, that you will see from now on. From the initial screening assays, Corema album leaf, this one here, was the one showing the best activities. Therefore, the results I will show you from now on are all related with this species, Corema album leaf polyphenols. Okay? So, Corema album leaves, commonly known as Portuguese crowberry or camarinha, as you may know it, it's endemic from Peninsula Iberica, and it was really poorly characterized. In fact, in this study, we performed the first chemical characterization of the polyphenols in its leaves, and we found by LCMS that it is mainly composed by myristin and epicatechin derivatives. To study the protection of these polyphenols, we used a very useful and versatile model, the yeast PD model. This is a model organism, and it's used to study Parkinson's disease because by expressing alpha-synuclein, that protein that misfolds and aggregates, under the GAL promoter, we can mimic the main pathological hallmarks of Parkinson's disease in yeast. Yes, sometimes people make fun of this, oh, do yeast shake? And I say, no, not with that purpose. Not with that purpose. So by switching the medium from galactose to glucose, we can do the switch on or switch off of alpha-synuclein expression very easily. As you can see here, when alpha-synuclein is expressed here in this line, the growth of the yeast is compromised in comparison to the control cells, this one, that grow very well. Surprisingly, when we treated the cells with alpha-synuclein, when they are expressing alpha-synuclein with the polyphenols, we see that although they present a longer lag phase, they recover growth very well, reaching the same levels as the control strains. So we wanted to understand why is this happening. Of course, we used several methods here. I'm just showing you this one. And we wanted to understand mechanisms underlying this protection, what was happening within the cells. So the most obvious thing was to look at oxidative stress, because oxidative stress is another central role, has a central role in Parkinson's disease and also alpha-synuclein misfolding and aggregation. So for this, we used flow cytometry and used two different probes, or dyes. One for general reactive oxygen species, ROS, and another one that it's more specific for superoxides, which is the radical that it's mainly produced by the mitochondria. As you can see here, when alpha-synuclein is expressed, both the general oxidative stress of the cell and the superoxide levels increase. And surprisingly, polyphenols were able to reduce both the levels of general oxidative stress of the cell and the superoxide levels. Went a bit further and studied this protection against oxidative stress in a human neuroglioma cell line, neuronal cells. We treated cells with hydrogen peroxide, that it's a known inducer of oxidative stress, and by flow cytometry using propydium we analyzed viability. And propydium iodide is a dye that only enters in cells who has lost membrane integrity, so it will label only dead cells. And you can see that after treatment with polyphenols, the percentage of dead cells decreased, showing that the polyphenols reduce the toxicity of alpha-synuclein by reducing oxidative stress both in yeast and in human neuroglioma cell lines. But we weren't happy still, so we continued to use the SPD model, and by fusing the alpha-synuclein to a GFP, a green fluorescence protein, we were able to look at the formation of alpha-synuclein inclusions. And in here you can see, I don't know if you see well, but here you have two cells with alpha-synuclein in the membrane and also in the cytoplasm, and in here you have these little bright dots that correspond to the alpha-synuclein inclusions, the aggregated form of alpha-synuclein. And once more, polyphenols were able to reduce the percentage of cells with inclusions. So the next step, we looked at the expression level of the protein, if it was being changed by the polyphenols, by the PEF. And we didn't observe any difference. The levels of the protein are the same without or with polyphenols. So at this point, we got intrigued and wondered, well, perhaps they are changing the clearance or the degradation of the protein. To do this, we used a very simple assay where we changed the galactose from glucose and inhibited the gal promoter, so there was no more alpha-synuclein expression. We stopped the synthesis to study the degradation of the protein, how it was being degraded. And here you have the results. At 0 hours of clearance, or in galactose medium, where alpha-synuclein is expressed, you have the same levels of alpha-synuclein. However, when you stop the synthesis by changing the medium to galactose to glucose, you see that after the treatment with polyphenols, the levels of alpha-synuclein are reduced. They are lower when cells are treated with the polyphenols, suggesting that polyphenols were promoting the degradation or the clearance of alpha-synuclein. So the logical step was to look at the degradation pathways of alpha-synuclein. In which pathway were the polyphenols interacting or interfering? Alpha-synuclein can be degraded by two main pathways. The ubiquitin proteasome system, that was my first scientific passion, I must say, where alpha-synuclein, it's tagged with ubiquitin, and then it's recognized by the proteasome and degraded, and you see here the fragments. Or it can be degraded by another pathway that it's autophagy, it's a bit more complex, and in this case alpha-synuclein is involved in a vesicle called the autophagosome, and then it is fused with the vacuole in yeast cells or the lysosome in mammalian cells, and the cargo is degraded there by the enzymes that exist in these compartments. To study the proteasome, we use the GFPU, which is a GFP, a green fluorescence protein, that has a tag that makes it constitutively degraded by the proteasome. So by measuring the fluorescence or the levels of this protein, we have a measure of the functionality or of the impairment of the proteasome. And you see that when alpha-synuclein is expressed, there is more GFPU, indicating that the proteasome is being impaired. However, and for my sadness, the polyphenols did not reduce this impairment by alpha-synuclein. So we looked at the other pathway, at autophagy. To analyze this is by Western blotting, we used the GFP ATG8 processing assay, where ATG8 is a protein involved, a very important protein in autophagy, involved in the autophagosome formation. And it is fused to a GFP, and when the cargo here enters the vacuole in yeast cells and is degraded, the GFP is cleaved from the ATG8 GFP construct. So we obtain this band here that corresponds to the free GFP. This means that by measuring the levels of free GFP in relation to the total signal, we have a measure of the functionality of autophagy. Not only the activation, but the functionality of autophagy. And you can see that when alpha-synuclein is being expressed, there is no free GFP, indicating that autophagy is being impaired, and you can see that the flux is reduced. The polyphenols promoted autophagy to the basal levels. It was the vesicles, autophagy vesicles, to the levels of the control, and this was very, very interesting. Rapamycin was used here as a positive control because it's a known inducer of autophagy. We also studied autophagy in human neuroglioma cells. This is a different assay, also by Western blotting, but in here we analyzed the LC32 levels, which is the homologue of ATG8 in mammalian cells. And we measured by using inhibitors of the autophagy of the lysosome enzymes, we were able to see the accumulation of LC3 a long time, and therefore analyze the autophagic flux. We measured this in control cells, cells expressing wild-type alpha-synuclein, or in the SYN-T aggregation model, where cells are transfected with a synuclein that has a truncated GFP, and also synphilin 1 V5, that it's a known stimulator of alpha-synuclein aggregation. And I have some pictures here. Here is the SYN-T aggregation model, where these dots here, and also these bigger dots, represent the alpha-synuclein inclusions. This is represented by the ratio of PEF and vehicle-treated cells, and also in human neuroglioma cell line, we observe an increase in the autophagic flux promoted by the polyphenols in alpha-synuclein wild type and also in the SYN-T aggregation model. Importantly, this was also independent of the alpha-synuclein expression levels. So at this time, we knew what was happening to alpha-synuclein toxicity, to oxidative stress, to the clearance of alpha-synuclein, to the aggregation of alpha-synuclein, and also to the autophagy and the proteasome function. But what was happening to alpha-synuclein itself? What was polyphenols doing to alpha-synuclein itself? To study this, we needed to use an in vitro assay, where in a test tube we only have alpha-synuclein and polyphenols, nothing else. Well, buffer and everything as well. And we analyzed the alpha-synuclein fibrillization a long time. For this, we used the THT fluorescence assay, where THT is thioflavin T. It is a dye that binds to beta sheets. So by using this, we can see the formation of beta sheets when alpha-synuclein is alone and represents its fibrillization. This is a typical fibrillization curve of alpha-synuclein. And when alpha-synuclein was incubated with polyphenols in 2 different concentrations, we saw that there was a reduction of alpha-synuclein fibrillization, an effect that was more visible with the higher concentration of polyphenols here. This was very intriguing, and we tried to study the nature of the species being formed by SDS page, where the samples acquired at these time points were run in an SDS page. And this is the normal blot we obtain for alpha-synuclein alone. And with polyphenols, we see that besides alpha-synuclein, we have the stabilization of this oligomeric species, or high molecular alpha-synuclein species in here and in here. So this suggested that the polyphenols were promoting the stabilization of alpha-synuclein oligomers. To study the toxicity of this species being formed, we incubated H4 cells with the species, with alpha-synuclein alone or the species formed in the presence of polyphenols, and measured the LDH release. It's a method for viability. And we were very surprised when we saw that the species, the alpha-synuclein species formed in the presence of polyphenols, were not toxic, in contrast to the alpha-synuclein species alone. So this figure shows us that the polyphenols are inhibiting alpha-synuclein fibrillization, promoting the stabilization of SDH-resistant oligomeric species, and these species are not toxic. To sum everything up, we know that alpha-synuclein, under pathological conditions, misfolds and aggregates, forming toxic alpha-synuclein species. These species will lead to the impairment of the mitochondria, leading to oxidative stress, and also to impairment of the quality control systems, the autophagy and proteasome function. That ultimately will lead to toxicity and cell death. The polyphenols are able to reduce this toxicity directly by inhibiting the fibrillization of alpha-synuclein, the formation of toxic alpha-synuclein species, and promoting the stabilization of the non-toxic oligomeric species, and indirectly by reducing oxidative stress, mainly superoxide levels, and also by promoting the clearance of alpha-synuclein by recovering the autophagic function. The shorter take-home message is that polyphenols protect from alpha-synuclein toxicity by reducing oxidative stress and promoting the clearance of alpha-synuclein through autophagy. This is the main important thing from this work. This opens new insights into the molecular mechanism of polyphenols and also new venues for drug targeting in Parkinson's disease. I would like to acknowledge the Disease and Stress Lab Biology team, the Cell and Molecular Neuroscience team, of course, my supervisors, the James Hutton Institute, where we perform the chemical characterization, FCT and Newberry for funding, the organizing committee for this lovely event. Let's hope to be here in 10 years, 20 years, 40 years, 50 years. And of course, thank you all for listening.